Hello dear students, welcome back again to Chinmoy's YouTube channel. So I hope you already have gone through yesterday's episode of reproduction in plants. So today it is the reproduction in plants part 2 we are going to discuss. So in yesterday's episode, if you have already gone through, I've discussed about two types of reproduction, that is sexual and asexual reproduction. In asexual reproduction, we have taught about the different types of asexual reproduction, budding, fragmentation, etc., spore formation and all. We have discussed about the vegetative propagation, that is vegetative propagation by natural methods. We have discussed about tuber, rhizome, corm and bulb, and by aerial method, by root and by leaves we have already discussed yesterday. So let us start with today's episode where I am going to discuss about the vegetative propagation by the artificial methods. So let us start today's episode. Now, vegetative propagation by artificial method, it's actually vegetative propagation. We all know it's a faster method, right? It's a faster method of reproduction. Hence, it is actually used in gardens and nurseries. So, it's very much faster than handling a seed. The growth of a plant from a seed, it's much slower process than this artificial method of vegetative propagation. So the methods that have been adopted by human beings for vegetative propagation are called artificial propagation. So the name given to this type of vegetative propagation is artificial propagation. Now in this episode we are going to learn about all the types of artificial propagation we have adopted, right? So please do subscribe to my channel if you want to know regarding this lesson and more regarding your subject, your topic and whatever I am teaching as per ICSE curriculum of 8th standard, right? So stay tuned, stay updated with my channel. Now in vegetative propagation, the first artificial method we have taken here is cutting. So what is happening here? In this method, we have seen a part of the stem with an axillary bud. Now it's being cut here. So part of the stem is cut from the healthy plant and is planted in a moist soil. And over a period of time, from this part cut down, roots develop at the lower end and it begins to grow into a new plant. So also new leaves, we can see that new leaves are also developing from that cutting only. So cutting is extensively used by the gardeners. Now we have seen that plants like rose. So rose, it is being done by the stem cutting process. Now there are cacti plants also, they are being done by this. Then we can say that bougainvillea. So bougainvillea is also a plant which is actually done by stem cutting process. So a part of the healthy plant, a stem is planted in the moist soil. So over a period of time we can see that new leaves are growing from that part only and new roots are also growing. So this roots and leaves, as they are growing, these part they again develop into a new plant, and it's quite a faster process than from the seeds, plants going from a seed. Now next we go to the next method. So the next method here is layering. This is actually a different type of method. In this method, what is happening? You can see that there is a branch in this plant, right? A branch is actually bent and it is covered by some soil. So the branch is bent and it is covered by some soil on top of it. Now after a few days, what we can see that roots are developing from this branch only, which have been covered by the soil. So under the soil there are roots which are developing and the branch is then cut off. This branch is then cut off, and hence we can see that from these new branch which was under the underground and from which the roots were growing, from there only we can see that a new plant is growing. Now what are the examples? What are the plants from like this layering is adapted in the reproduction methods, vegetative propagation? So the main plants which are being done in this process are lemon. First we can say that lemon. Rose is also done at times by this layering, that is bending of the stem, and it is covering by soil, and then roots grow from that part, and the other branch of the plant is cut off and the new plants grows from this plant only. Now lemon, rose, jasmine, these are the few examples of this layering. They are grown in this manner. Now next to this comes the grafting. Now here see grafting. Grafting is also a new type of method. Now it is a method when different plants are combined to obtain a plant of superior quality. So this is a different plant and this is a different plant. So different plants are obtained with superior quality. So a stem cutting called scion. So these stem cutting is called the scion. From one plant is attached and tied and rooted stem with the stalk of another plant. So this is the root stock of another plant, and this is scion of one plant. So how they are combined? They are tied with one rope or something. So after a few days, what we see, the stalk and the scion, they merge together, and by the rapid division of cells, and bulb develops, and the stalk supplies essential nutrients to the scion. This stalk is supplying essential nutrients to the scion, the upper part, and this grafting develops new varieties of and features of two plants. So it is usually done in plants such as mango and rose. So two important plants which are being done by grafting method is mango and rose. So two different qualities of mango. So one part scion has been cut and grafted, another is root stock of another plant. So you can get a new variety of mango or a new variety of rose. So in rose it is done to get flowers of different colors, like yellow and red might be combined, or white and red might be combined, different colors, and it is done usually in mango, so that you can get different varieties of mango. So these are two types of plants. Mostly in them only we have used this grafting techniques. Rather these in gardens and all, they have used this technique to produce new type of mango, new quality of mango and rose. So let us move on to the next type. Now here comes tissue culture. So what is tissue culture? Now it is a method where plants are they are grown artificially in test tube under several lab conditions. So what is done in this technique? This technique is actually called micro propagation. So this technique is having another name. It is called micro propagation. So this is having another name called micro propagation. So a small piece is cut off. So what is done here? A small piece is scraped out or is cut off from the tip of a plant, and now this tissue is actually grown in a suitable nutrient medium. Now it grows in an unorganized mass, and so that is called the callus formation. So this tissue sample is placed in a agar growth medium. So there are several different growth mediums in which this tissue is being put into. So containing nutrients and auxins. Now auxins are the plant hormones, okay, and nutrients, several nutrients which are required for the growth of this tissue has been put into this petri dish. Now samples develop into tiny plantlets. So what are plantlets? They are mini plants. So samples are developing into tiny plantlets, and these plantlets, they are into compost, right? Now small parts of this callus, we are telling that they are put into another medium containing the hormones. They are put into another mediums containing the hormones and forming the plantlets. So these plantlets are then transplanted into, as these plantlets are formed, they are then transplanted into different soils, pots, to grow into individual mature big plants, right? So there are unlimited number of plants and can be produced, because from this tissue culture we can produce a huge number of plantlets rather. And this technique is used in the propagation of generally ornamental plants, or generally different kinds like orchids. They are ornamental plants, we all know. We can also tell about asparagus. So all these ornamental plants are being produced by this tissue culture method, right? Now we can say that the mention may be made of chrysanthemum. So these are the plants under this tissue culture. These plants are being produced under laboratory conditions. So is it clear to everyone? A part of the plant sample is taken. They are being produced. They are being taken in a petri dish with a suitable hormone, nutrients and all in agar growth medium or any other growth medium, where the samples develop into plantlets, and these plantlets are then planted into soil or any pots where they grow into mature full plant, right? So this is all about tissue culture. Now we are going to discuss about the advantages and disadvantages of this type of propagation. Now first we are going to discuss about the vegetative propagation advantages. So what are the advantages of this method? Now this is a faster method, I have told earlier also. Rather than growing from seeds, they bear flowers than those produced from seeds. Now extremely these are useful for the production of seedless plants such as banana, grapes and sugarcane. So there are many plants which are totally seedless, grapes, banana and sugarcane. So for them it's very much essential that they are grown by the method of vegetative propagation. Now new plants are exactly the same as the parent plant and can survive in unfavorable conditions also. So if the new plants were having different characters from the parent plants, it would have been difficult for them to grow into exactly new plants because their survival rates reduced. But in this case, as the new plants are exactly the same as the parent plant, there are 100% chance of their survival if the condition is unfavorable also. So what are the disadvantages? The disadvantages mainly, that is, all the offsprings have the same feature. So all the offsprings are having the same feature, and so they are prone to infections which can result in the destruction of the entire crop. So over a period of time, newer generations of plants are produced via vegetative propagation, and they lose their vigor. They lose their vigor, they lose their strength, and there are chances of overcrowding as there is no dispersal. So there is no dispersal of plants, they are growing at the same place. So this vegetative propagation, they can cause chances of overcrowding also, as there is no dispersal in these plants. So is it clear to everyone? So these were all the vegetative methods of artificial methods, vegetative propagation by artificial methods. So today we are here completing this lesson, that is the asexual reproduction I am completing. So tomorrow I'll be there again with the another part, that is sexual reproduction in plants. So then we'll be completing this lesson into 3 or 4 episodes. So please subscribe to my channel, stay tuned and stay updated with whatever lessons I am posting as per your curriculum, so that you can study this lesson in a sequential and systematic manner. So go through my episodes, what I am teaching, and don't forget to subscribe. Thank you.